I'm going to quickly go through another assay we've been developing for standard STR analysis. Before I start, is there anybody in the room who's not familiar with short tandem repeat assays in general? I was assuming a lot of people here do forensics analyses and are familiar with short tandem repeats.
Yes, they should be.
Okay, so I'll just proceed. Well, that way I'll go straight into it. Okay. We've been developing with partially with support from the NIJ an assay to do short tandem repeat analyses using the standard core CODIS loci. That's same loci that make up the convicted felon database, for instance, that has 6 million profiles in it now. I'm going to go through the format of our assay, a little bit of detail about how we design primers, how it works with respect to a database, the layout, how a sample is analyzed using our system relative or compared to traditional typing. And what's become very interesting, I'm just going to introduce it, and then John's going to go into what it actually means and what's interesting about this. There are a lot of polymorphic alleles out there. There's a lot more alleles than you see with traditional typing. And I'll just show you what the data look like. So right now, we're focusing on the 13 core loci that make up the CODIS database, or that go into the CODIS database, and then amylogene and the sex type. And as you know, they're on different chromosomes, so you get a high differentiation statistics because they segregate independently during meiosis. Using the technique that we're using with mass spectrometry, it's very straightforward to do STR analyses. You can design primers basically anywhere outside the repeat, and you can use a published repeat structure and develop a database of known base compositions for what you expect. What this means is we don't need an allelic ladder. We get a very precise measurement. We measure an intrinsic property of the amplicon that tells us what's the base composition, what's the base composition that's expected for, say, allele 13 of this locus. We can compare our result to that directly. We don't have to run a standard next to it. And then we do a little bit of multiplexing with this. So what we'll see here is if we design primers to a particular locus, this example is D13, and then we run an assay, any assay that gave us base composition, or if we sequenced it, for instance, but in this case we're running mass spectrometry, we can take the output for any given reaction, and then we can compare that to a database of known alleles or expected alleles. Here we see we have alleles 5 through 13 sitting in a database, and we know the compositions that would result from PCR reaction that had one of those or both of those in there. Our layout for the assay is essentially the same as our mitochondrial assay. We have 8 reactions, so it fits in a column of a plate. Each of 5 reactions has 3 primer pairs in it, 3 different loci together. It's minimal multiplexing, as it's, compared to, say, the PowerPlex 16, we don't do them all in one reaction. There is redundancy between reactions for some of these. So you'll see here D3, or wait, excuse me. Where's the one I was looking for? Here, D5 is in both of these reactions. So you get a little corroboration between reactions, so you're not mixing samples, you're getting the same result between them, since we do split them up into multiple reactions. And then we have 3 loci that the upper limit of the length on these, we put our primers as close to the repeat as we can get them. But D21, FGA, and D18 have a fairly large upper limit on, especially FGA with some of the rare alleles. And those can get up to, say, 2 or 250 base pairs, even maybe 260 for some of the rare ones. that's pushing the limits of what we can resolve with the mass spectrometer. We're finding out we can actually do it if we don't try to multiplex them, though. So we keep these ones by themselves. We don't put other primers with them. If you put too much into the mass spectrometer together, you'll get a lot of congestion. You get a lot of peaks, because you saw the reactions I showed for the mitochondria with only 3 primer pairs and all those peaks. As products get bigger, each product has more peaks too, because it has many more. Well, Steve showed an example earlier with one that was like 300 bases long, and there's a lot of peaks in there. So we have a standard setup then that's similar to the mitochondrial layout. You can split one sample across the 8 wells of a column of a plate. Same type of kitted format. We manufacture a plate that you add 5 microliters of sample to the plate and you put it in a thermocycler. This is an example of what some of the data will look like coming out then. Similar to the mitochondrial assay, the only difference is, well, there's 2 chromosomes for each locus now, because mitochondrial is haploid, but this is diploid, so you'll see 2 products per locus, or 1 if it's homozygous. And the difference between this and what you might be used to seeing in a CE is you'll get 2 peaks per product rather than 1, because the strands are separated from each other. From that, though, we can take each of those, we get their mass. This is just one locus. There's another, you can see that this part of the spectrum, this is a multiplex assay. There's another locus in there, I haven't labeled it yet. This one's D13, that's colored blue. These 2 strands make up a composition, A32, G11, C34, T57. This is another one that's just longer. If we look in our database, then we can find 32, 11, 34, 57 right there with no ladder. It's just a direct measurement. And then here we have a 13. So we have an 11 and a 13 for this sample. Here's the other one in that same reaction, VWA. There's another one, but it's off the screen. Same thing, 2 products. There's a database, there's more alleles in it here. 17 and 18 together. It's very straightforward. There's an interface similar to the mitochondrial interface, except with some differences, because it's a different kind of analysis. You get kind of a cartoon of the alleles that have been assigned, a summary of them. I'll get into this in a minute. You'll see that these look different. This one says 15 with a G to A polymorphism relative to the reference. And there's a few of those. You'll see a lot of them actually as go through the data. Same thing as mitochondria, you get a bunch of, you can do 12 on one plate. Usually 10 though with a positive and a negative control. A summary of allele calls, it's going to look very familiar. It's just a profile. So there's 10 samples here. Actually this one has 12. No, 10, positive and negative. Now compared to typical typing that might be done on ABI instrument, for instance, with capillary electrophoresis, where you can get significant throughput because you can do 96 parallel reactions on some of the higher priced instruments, that relies on just the size of the product. So you get an allele number based on the number of repeats that are in that product, right? And you have to, since you're measuring mobility, you're not measuring an intrinsic property of the product, you have to compare it to some standard. So before you run, you've got to run an allelic ladder, and it's generally recommended after you run so many, you might run the ladder again to recalibrate, right? So anywhere from each run of 10 or so. Takes about, and I've heard you can do these faster, but recommend about 25 minutes per sample. People tell me you can run them in about 15, depending if everything comes off by the time you're done. It's highly multiplexed, at least from Promeg, you can get 16 all CODIS loci plus a couple extras in one reaction. And there's a pretty large dynamic product size range because you can keep running the capillary. In comparison with the mass spectrometer, you get a much more precise measurement of what you're looking at. And what we're finding out, first advantage is you don't need an allelic ladder. You're measuring a property of the product. So you measure its mass, you get its base composition. It turns out that there's actually quite a few more alleles than you might expect just based on the length. They're not all the same just because they, if they're 180 base pairs long, they might have a different composition. Our current format is 8 wells per sample. That's roughly, when you take into account once the machine's running, our machine can run one plate and be cleaning another plate as one's spraying into the mass spectrometer. So you get some parallel, you get some time benefit from that. If you're running at least 2 plates, one sample will take about 7.5 minutes. That's somewhat comparable to about a 4 capillary genetic analyzer when you consider you can run one sample and get the whole thing. Our multiplexing is limited compared to typical typing. We can't do all 16 loci or 14 loci in one reaction. And our product sizes are limited. We can go up to about 250 in a single plex for an STR as long as we don't need a de novo base composition without knowing anything about the product we're looking at. But when we do an STR, we know it's a very limited set of possibilities. We know the allele structure, we know the repeat. We can say we have something that's 18 repeats long, and it's a C to T SNP away from what we'd expect from the reference, even at a pretty big size. Now, what we noticed right away after starting looking at running STRs on the mass spectrometer is that not all alleles are exactly what you'd expect relative to a reference. Many of them actually have polymorphisms in them. Some of them are within the repeat, and some of them are in the flanking region, or regions. And there's actually quite a few more alleles out there than, they're not rare. So here's an example of D5, where we just do a standard measurement. We get products, see base compositions from them. This little asterisk just points out that if you went back and calculated using an algorithm that looks at the masses of A, G, C, and T, it wouldn't line up exactly to these masses because we're using a carbon-13 enriched. A carbon-13 enriched GTP, it's about 10 Daltons heavier than a normal G. That changes I'll go into that in the software talk later. Here we have a database of alleles for this locus. And we see an allele 10 there. There's the composition. And then we look for this one. We don't find it here. See 5634, there's no 5634. There's a 56, there's a 10, but then there's a 35 and a 48, and that doesn't line up. But you do notice right away it's an allele 12 that's got 12 repeats, but it's got a base switch in it. So it's actually an allele 12 with a G to T SNP. And then you could see, well, what would happen if there was an allele 12 with a G to T SNP in it mixed with an allele 12? They would both be there and they'd separate from each other and you'd see them. And that's hypothetical and that's a sample. This is one of the samples that we got from John. And we ran some and we see for this individual had an allele 12. Matches the database. It's not just a naming issue, in other words, they're both there. This person is heterozygous. Whereas if you did a CE, you'd see a homozygous profile. Now, when we look at for this particular locus, same locus shown here, at the different samples we've run, we've seen each of these. There's G to T, there's an A to C. So there's 3 different allele 9s here so far. I mean, there might be others, I don't know. A couple allele 10s, and various polymorphisms. The same polymorphism tends to occur in incrementing repeats. So you'll see an allele 10 and 11 and 12, but then you'll see a G to T in the 10, and you'll see a G to T in the 11 and the 12, just like you'd expect. And some of those are, it might be AGAT, and then somebody else might have AGGT in one of the repeat units. The AGATs that are repeated over and over will slip back and forth in replication just like you'd expect, creating length differences through evolution. And that AGGT will just be there for the ride, and it'll show up in the different lengths. Now we've seen running, we did some samples with John Butler, 95 reference samples. These, oh, I don't have the reference down here, I apologize, but they're part of the reference set from Caucasians, African Americans, Hispanics that was published by NIST. It was a larger set, this is a subset. And then some 590 or so reference samples from Texas, from John. And we've seen polymorphisms now in 11 of the 13 core loci. Some of them are very rare. We don't see them very much. We've only seen a couple here in CSF. But in 2 of the loci, they're very frequent. And in each case where we see them, we've seen up to 10% almost same length heterozygous, where they wouldn't be seen as heterozygous if you couldn't tell the difference between polymorphic alleles. And some of these are actually just, it's arbitrary which one was called the reference at the time the allele structure was used to design a database, because here we have more than half polymorphic. We could just turn that around. But in each case, we've seen both the reference and the polymorphism. So it's not just a naming thing. Now, if you look just at the allele base, and John will go into what this means, but it's kind of interesting. For several of these, here, D21, we've seen 24 additional alleles. I mean, these are just different alleles in the population. They can be used to differentiate people just like length. And they're stable. You can examine a pedigree, and you'll see that somebody who has a 15 with a GA will pass it on to their offspring. We've seen quite a few of these. This just shows the number of discrete alleles have been observed so far beyond the published, or what you'd predict just from the published structure. And there's been several published reports too where people do sequencing. There's some sequencing that's been generated from D21, for instance, and VWA. There's already a lot of known polymorphisms out there. This is an example of what the data look like for one sample, for each of the reactions of one sample. So here we see D3 has 2 GDA SNPs within allele 15. And that individual happens to be homozygous for that. They have 2 copies of that one. And then these are just standard alleles. And then here there's no polymorphisms in these 3, just a profile. Here again we see a 12 with an A to G and a 16 with an A to G. A to G is common in this particular locus. Here's D5 again. That was in the 2 reactions before, so it's redundant. There's D21, so you'll see it's by itself. It's a singleplex reaction. FGA, D18. It's very similar to looking at gels, just 2 peaks per product instead of 1.
Did you see any stutter peaks?
We have seen stutter peaks, yeah. In fact, there, I think we're probably seeing a stutter from, I'd have to look at the masses on that. Here, and then here, probably stutter from 2 different products. We see, it depends on the locus, just like for traditional typing, but yeah, you'll see stutter. summary, we've so far discovered the core CODIS loci, have a standard assay layout. We're working towards getting this into a commercializable final product. We're still in development. But there's a lot of polymorphic alleles out there. Right now, the sensitivity per reaction is comparable to, say, like a Promega kit. Between 1 and 200 picograms, we recommend up, closer to a nanogram. But we have 8 reactions, so that's something to keep in mind. Okay, questions? Yes, Les.
Do you always find with your repeats with the SNPs in them, do you always find equal relative intensities and relative abundance between the non-SNP variant and the SNP variants?
They're not always perfect, but they're not always perfect anyway.
Yeah.
So no, there isn't any noticeable difference between SNPs and none. They're just another, the reference is arbitrary anyway. You call one the reference and the other one would be a SNP backwards SNP from it. So we haven't noticed any particular.
Any degree of variation between the two?
No, not really.
With your summary slide, is that, you were showing your sensitivity, is that how much you're targeting per reaction? You said you put 5 microliters into your plates. Is that what you're targeting, the 100 to 200 picogram range?
Per reaction,
And
so times 8 per sample.
If you have a lot of excess DNA, does that affect your sample like it would in typical STRs that you'd have to re-amplify the sample?
It doesn't look like it at this point, but we haven't examined that in a mode that I could answer that question thoroughly. So far, it doesn't seem so. We don't need to quantify, for instance, you just run. And with the mitochondrial assay, which is very similar in nature, goes through the same analysis. We've gone up to 37 nanograms per reaction without an apparent effect. But I can't answer that thoroughly because we haven't done that analysis. Yes?
Is there any possibility that a misincorporation for PCR could result in the appearance of a SNP?
Anything is possible, except we do replicates of the same sample and we see the same result, so it's reproducible. And you can look at a pedigree where you see transmission from parent to offspring.
I'm just saying in casework where you may not have the ability to look at a pedigree. But you're saying you've done samples in...
Yes, and we also have several of these loci are in 2 reactions. So you see the same result.
The same principles apply that if you put too little DNA in there, it could happen. That's why you have to have a minimum amount of DNA. Otherwise, the PCR is the same.
Well, you'll see a severe imbalance too if you get too little DNA at some point. It gets kind of random.